So what you're looking at here is a motor neuron. So this is in the individual who dies from ALS. The problem is that in the spinal cord, the individual motor neurons are dying. And when we look at it through the pathology, what we see is this is a very large cell, which is what we expect to see. This is where the nucleus to the cell sits. And then this area here, this sort of rock-like material, very dense aggregate, should not be there. And this is the new protein. This is what we call RGNEF, the one that we discovered. And you can see as well there's material of it sitting all throughout the cell. It should not be there. And in confocal microscopy, we have the ability to turn the cell around and to look at it in 3 dimensions. But then we can look at it with a second protein. So this is the same cell looking for yet another protein, which we know to be in the same family of RNA binding proteins. And here we see it as a rock as well, and we see all of these we call aggregates or skein-like structures. This is not normal. You never should see this within a cell happening. So the question becomes, we have the same cell, 2 different proteins pathologically occurring. Are they together? So we can superimpose those 2 images, and wherever they overlap, you'll see them showing up as being yellow. But all that tells you is that they're in the same neighborhood. It doesn't tell you that they're absolutely together. So what we do, and this is part of the capacity we have over at the Robarts, is to subtract away all the background noise and just ask the top 2% of where they match. We're going to assume that those are actually together. And then we go on to prove that. But what you'll see next is white. And so the white is that top 2% of colocalization. So you can see throughout the entirety of the cell, these 2 proteins are together. Now, to make that easier to see, what we do is get rid of everything else now. So we'll get rid of all the background, and what you're left with is the skeleton of that cell showing everywhere within that cell. Remember, the nucleus was up here. Now we've got this massive aggregate here and all of these skein-like structures throughout the cell. These 2 proteins are located together. Right? Nobody had ever seen that before. This is brand new.